Hello, everyone, and welcome to today's webinar, a roundtable discussion in which we'll be talking about getting started with mRNA-based therapeutics, myths, considerations, and characterization. This event is being sponsored by TriLink Biotechnologies. Hello, everyone. I'm your moderator, Kevin Davis. I'm the editorial director for Genetic Engineering and Biotechnology News. The author of Editing Humanity, The CRISPR Revolution, and the New Era of Genome Editing, out in paperback, somewhat relevant to today's conversation. And I would just briefly mention one of the 100+ journals published by Marianne Liebert Inc., Gen Biotechnology, the new marquee peer-review journal, publishing original research in all the areas featured so prominently in Gen magazine, including RNA therapeutics, cell therapy, and many of the other key topics that you'll be hearing about in today's discussion. Please review our recent issues at www.genbiotechjournal.com. Okay, so in today's roundtable, we will explore mRNA challenges, including usability, stability, and technological limits. We'll hope you'll gain insights into developmental considerations for clinic-bound constructs and better understand the crucial role of characterizing mRNA in the early stages of development. My three guests will do their best to dispel some myths around mRNA challenges in therapeutic applications, share their insights into mRNA construct development for clinical use and developmental considerations across stages, and consider the importance of mRNA characterization early in the development process. And so to do this, I'm delighted to say I've been joined by an outstanding panel of scientists from TriLink. Hannah Ellerbrock is the Director of mRNA Manufacturing at the company. Ben Hudson is Associate Director of mRNA Research and Development at the company. And Ina Kukareva is the Director of Analytical Services at TriLink. Just before we get started, a quick note about questions. We invite everyone in the audience to submit questions during our conversation. Our speakers won't be able to address those questions live today, but your questions will be read and evaluated by a TriLink scientist or representative who will be doing their best to reach out back to you to provide an individual answer or response in the days following the webinar. So to get started, I thought we would just go round the table, so to speak, and invite everyone to just briefly introduce themselves and in their own words, say something a little bit about their background and their current role at TriLink. Ben, let me start with you. You're the top left on my screen.
Sure. First, thanks for having us, Kevin. It's a great pleasure to be here.
Yeah.
I am a PhD and postdoc trained RNA scientist. I've been doing this for a long time, and I'm thrilled to be at TriLink. I've been here for about 3 months now as the lead of the R&D biology team, the mRNA team. So it's a great pleasure to be here. I've been studying RNA my whole life, and it's exciting to see its prominence that it deserves.
Very good. Hannah, welcome. Tell us a little bit about yourself.
Hi, thanks for having me. I'm Hannah Ellerbrock. I've been with TriLink for about 4 years now, holding various positions across both GMP and research grade manufacturing applications for mRNA products. I have a master's in bioinformatics and am really passionate about the future of mRNA therapies and personalized medicines.
And Ina, hello, welcome. And would you like to say something about yourself, please?
Thank you so very much for having me at this webinar. So my name is Ina Kukareva. I am a Director of Analytical Services, and my role is to oversee the development of new and optimized, mostly physicochemical analytical procedures for the analysis and characterization of products made at TriLink, ranging from small molecules, such as MPPs and clean-up reagents, to oligonucleotides, to large nucleic acids, such as mRNA and DNA. My background is analytical chemistry and synthetic chemistry. I am a synthetic chemist by training. And first, I joined TriLink Biotechnologies in 1999. Yes, I have rather long history with TriLink and contributed to the company for 12 years, first in manufacturing and later as part of R&D team. Prior to rejoining TriLink, which I did in 2019 as Associate Director of Analytical Development. I was working as part of CMC analytical development team of the 3 pharmaceutical companies, Nitodenko, Arcturus Therapeutics, and Regulus Therapeutics. All of these companies were specializing in a nucleic acid-based therapies using LNP delivery platform, which gave me, I think, unique and broad experience with development of analytical methods for characterizing RNA, and that's why I'm on this webinar. Thank you so much.
Very good. Thank you so much. Great to meet all of you. So let me start with our first question. What are the critical components of mRNA and how do they impact manufacturability? Who would like to take that?
Sure, I'll take that first. Thanks. So as hopefully everybody knows or is learning and joining this webinar, mRNA is still a novel and nascent technology, but it's already a well-proven technology. I think the global pandemic has helped cement its place in history as helping really bring therapy to the forefront. So it has numerous applications in many different therapeutic spaces. At its most basic level, and kind of why it was such a success for the vaccine world, mRNA leverages the cell's translational machinery to amplify the amount of protein that can be delivered in an otherwise volume or concentration limiting way. It has the advantage of being readily reprogrammable and more rapidly synthesized than most cell-based protein productions. Can also be designed to work in multiple therapeutic modalities. And so for that, let's kind of go over the key components and we can talk about just how each part contributes to its use. So starting at the very 5' end, the very beginning of the RNA, you have what's called a cap structure. And the cap is used by the cell endogenously to identify, it has a couple of roles, but to identify self-RNAs and also to help identify RNAs that will be turned into proteins, so translation-bound proteins. It binds to translation initiation factors, and it comes in several different structures and flavors. In most cells, in mammalian cells, you'll call them cap 0, cap 1, or cap 2, and this just depends on the methylation status of the ribose sugar. So how many ribose sugars in that first part of the message are methylated. And for synthesis purposes, and we'll talk about this more later, caps can be included co-transcriptionally or added enzymatically after transcription. So lots of different options there. The next part that you'll encounter as you move down the RNA I'm going to lump these together, but it's the 5' untranslated region, and then after the coding sequence, the 3' untranslated region, and these sort of serve similar functions, so I'm just going to kind of lump them here. But these are stretches of sequence that are found normally in all RNAs, endogenous RNAs. They help modulate the expression and longevity of RNAs. They can have secondary structure, meaning they have hairpins or some binding events that the RNA binds to itself. They may or may not recruit translation initiation factors or help recruit other proteins. And they can vary in length quite a bit, but are typically, especially for therapeutic RNAs, in the order of 100 to 200 bases. During mRNA design, UTRs can be pulled directly from nature, such as the commonly used globin genes, or they can be designed synthetically if there's a really particular characteristic you're after, and that's going to be on a case-by-case basis. Moving downstream again, we run into the coding sequence, and the coding sequence or the open reading frame, is flanked by a start codon and a stop codon, so an AUG, and then a couple different flavors of stop codons, and that's read in codon triplets, so you have 3 bases in a row that tRNAs recognize, and then add your polypeptide chain. And these signals just tell the ribosome when to start and stop making the protein. Downstream of the 3' UTR that I already mentioned, you have a poly A tail. And this is, the tails are found in many types of therapeutic RNAs, but not all. And so we kind of wanted to make this broad in this discussion and say that, if you're making an RNA, not all of them are going to have the same types of tails, not all of them are going to have the same lengths. And so poly A tails, normally they help to recruit translation initiation machinery, poly A binding protein, for example. They can vary in length fairly dramatically. Some are quite short, ranging from 50 to 150 bases. And then there's even some variety for types of poly A tails. So you can have full-length homopolymeric runs of A's, or you can have segmented tails. Some vaccines have found success with breaking up the poly A tails, especially in manufacturability, where you'll have a section of A's and then it's broken up by a few bases and then another section of A's. And that brings us to the end.
Literally. Ben, let me stay with you. How do critical components of mRNA then impact performance?
Yeah, great question. So again, each of those components can have, it really needs to be tailored to the type of therapy and the goal that you're getting to. So to begin with the cap, like I said, it really serves to identify self-RNAs and also to recruit translational initiation machinery. So the cap has a big role in preventing rapid degradation, and it helps reduce immune reaction that you would get from putting in so let's say a 5' triphosphate RNA where you don't have a cap structure. Typically, you'll get a gigantic immune response, and then the translation will be shut down, and you won't get any protein produced. So if that's your goal, then you would want a good cap. The type of caps can all sort of affect that, and whether or not you add it co-transcriptionally or enzymatically. So cap 1 tends to lead to greater protein production than a cap 0 structure, or maybe a cap 2 structure might have lower immunogenicity than a cap 1 or a cap 0 structure. And another, for example, TriLink's most recent cap, what we call M6, leads to even stronger translation because it has this N6 methylation. And that, through mechanisms we're still exploring and investigating, increases protein production even further. The UTRs, their design can really vary greatly. They can dramatically increase or decrease mRNA longevity and translation. Some UTRs are designed specifically to recruit translation initiation factors, while others have a high degree of secondary structure, maybe to prevent nucleases and promote longevity. A modified bases, which we didn't really talk about in the first part, introducing that, but sort of can be included anywhere or in certain sections, depending on the length of the RNA. But modified bases are a big component of why recent vaccines have seen success. A couple of big ones that people have probably heard of. There's N1-methylpseudouridine, 5-methyl-C, 5-methoxy-U. These big players help to reduce the ability of the innate immune system to recognize these RNAs and say that they're not native RNAs. And so that allows the RNA to stick around longer, and that allows the RNA to make more protein if a vaccine is your end goal. Then something to think about for modified bases is that they may alter the secondary structure. So if your RNA therapeutic goal is to have a well-defined, say, a tRNA or an aptamer where you have a really defined structure that you're going after to bind a molecule, then having modified bases may affect that, and you might have to have fewer or more or really fine-tune that. And then to bring us to the end, the poly-A tail and how it affects the performance of RNAs. In general, I would say that slightly longer poly-A tails have been found favorable and have longer half-lives. So if you're trying to keep your RNA around a long time in the cell, then you would probably want a fairly long RNA. However, it's not always the case, and it's not necessarily borne out in the literature that all RNAs that have longer poly-A tails. So it really needs to be a case-by-case basis and how you investigate that.
Very good, very helpful opening remarks, Ben. Thank you so much. So I want to drill down now on the topic of manufacturability, if that's a word. I think it is. Hannah, let me turn to you. There are a lot of options out there. So how do the various components, many of which Ben has just nicely described, how do they impact manufacturability?
Yeah, absolutely. So starting with the synthesis reaction, you're going to have a major impact from your capping strategy, your coding sequence, and the UTR sequences that you decide to use. All of these components will change the performance of that synthesis reaction. And so you may need to optimize that synthesis to hit the yield, capping efficiency, integrity, and other key quality attributes that are appropriate for your program. Drilling a little bit more into the capping side of things, co-transcriptional caps can really simplify how you can set up that synthetic reaction. It's going to make your development requirements much more straightforward, and it's typically easier to scale that reaction than a solution like an enzymatic cap. Cap analogs also need to have very high capping efficiency to make your manufacturing process streamlined for something like a clinical application. If you have a cap strategy that results in a lower capping efficiency, it's going to complicate your downstream processing when you have to introduce solutions such as phosphatase treatment to cleave those uncapped ends. Additional enzymatic treatments like that mean more heating and processing for the mRNA. it means you have a more complicated clinical supply chain, and it means that you're going to have more challenges at maintaining the highest possible integrity of that mRNA coming out of your manufacturing process. Moving on to complex sequences, a common challenge that we'll see is repetitive or highly complex regions, especially with the UTRs. And I'll start with those UTRs. A complex UTR region can actually inhibit the initiation of your T7 polymerase during the mRNA synthesis. And so that may mean additional further optimization and longer study times to be able to get to the next stage in development. Repetitive sequences are going to result in longer plasmid lead times and some challenges in that manufacturability. Not only does that contribute to then an overall longer turnaround time for your final mRNA product, but it can also contribute to challenges during the mRNA manufacturing, such as early truncations. You can address some of those with purification strategies like oligo-DT. The good news is those are very scalable and ready to go into clinical manufacturing. So it's not so much a challenge at the mRNA stage, but you should expect longer lead times when working with something that's highly repetitive. Moving over to the poly-A tail lengths, Ben alluded to the wide variety of strategies that we can implement for an mRNA on the tails. Plasmid manufacturability, revisiting that again, you can track in an ADA tail relatively easily, and that's relatively standard in the field at this point. But sometimes you may need a longer tail to be able to meet your modality appropriately. Some of those would be 120As. There are a few strategies that you can go forward with on a 120A tail, such as PCR or introducing a linker strategy. Anytime you're introducing a new approach, like bringing in PCR to a tailing reaction, you may have some challenges when you get to a clinical level of production due to concerns around fidelity and manufacturability control in the process. Enzymatic tailing's another option that you can go with for your manufacturing process, but that's going to result in a more heterogeneous population of mRNAs with different tail lengths, which can be its own challenge depending on how tight you need that specification. Moving down to just kind of the final consideration that I think is good to bring up for manufacturability of mRNA is the robustness of your clinical supply chain. So it's exciting that we have so many new technologies and novel approaches to mRNA coming through right now. One thing to keep in mind when you're pursuing those is that if you're using key raw materials in the production of that mRNA that may not have made it to clinical production yet, your lead times for clinical manufacturing are going to be a lot longer as you try to find a good solution that's going to be compliant with requirements for your different stages. So we recommend to minimize reliance on raw materials that are rare wherever possible, and to avoid raw materials that typically involve the use of animal origin components to simplify your pathway into the clinic. You can never start assessing your raw material plan early enough in your program. So it's something to keep in the back of your mind through early stages of development.
Thank you, Hannah. That was great. I want to move on to Inna now. Inna, how do those critical components impact the choice of methods and analytical development?
Absolutely. So we just talked about the major structural features of the mRNA molecule: cap, tail, UTR, and coding sequences, and how these features may impact mRNA performance and manufacturability. These structural features, together with process and product-related impurities that may be present in the final mRNA drug substance, together fall into three major categories of the critical quality attributes, namely identity, integrity, and purity. Today, testing for these critical quality attributes involves a wide range of diverse analytical workflows. To confirm mRNA identity by sequence, which includes 3' and 5' UTR and coding sequences, there is now a large choice of sequencing platforms, ranging from good old Sanger sequencing to next generation sequencing platforms, such as, for example, Oxford Nanopore sequencing. mRNA integrity and identity by its size traditionally is determined by gel or capillary gel electrophoresis. However, HPLC and LC-MS-based workflows are becoming more and more popular as orthogonal techniques to further evaluate quality of mRNA. Thus, LCMS-based workflows are now commonly used to determine 5' cap structure and capping efficiency, to evaluate poly-A tail properties such as its length and distribution, and to perform mass spec-based sequencing/oligomapping. Product-related impurities such as truncated RNAs and RNAs without poly-A tail can be efficiently detected and quantified using IPRP HPLC method, while size exclusion chromatography can be used to monitor potential formation of mRNA aggregates. Now, among the most critical product-related impurities are double-stranded RNA species, accurate quantitative analysis of which still presents significant challenge for the scientist. Traditionally, these species are evaluated by immunoassays such as dot blood or ELISA. However, these days, researchers are actively exploring novel technologies to detect double-stranded RNAs, as for example, bioluminescence assay or cell-based bioassays such as recently reported by Promega. And finally, even though this goes beyond the scope of this particular webinar, there are also several process-related impurities such as free non-incorporated nucleotide, T7 polymerase, residual T7 polymerase, residual DNA template. For all these impurities, there are now well-established analytical procedures that can be found in many open sources, such as, for example, USP guidelines.
Great. Ben or Hannah, is there anything you would like to add to that otherwise very comprehensive response?
Yeah, I can talk about a little bit for the design. There are different ways, so specifically the process-related impurities. So there are ways to remove it from manufacturing, and there are also ways to sort of design around them. So when you're looking at how you want to design your process, there are IVT reaction conditions that may or may not be more optimal and give you different impurity levels, and those will all be determined after the fact in terms of how much you have to remove, but they can sort of be manipulated before the fact too. So optimizing from design to analytical process and manufacturing, I'm sure Hannah can add something too, but there's ways to modulate all of those attributes and impurities at the whole stage of the RNA process.
Hannah, you're nodding furiously.
Yeah, no, I would just add on here, I think, hit the nail on the head, but one of the key elements when you're deciding how to set up your manufacturing process and the development of your mRNA is defining these key quality attributes, what matters to your application the most, and then give yourself the best shot on goal to achieve success on those key elements.
Well, that's a nice segue to my next question, Ina. How does one go about choosing the right analytical method for a specific critical quality attribute?
This is an excellent question. You see, because therapeutic RNA are such a complex molecule, comprehensive analysis and characterization often require not just one or two methods per critical quality attribute, but I would say rather a collection of complementary and orthogonal analytical procedures. For example, capillary gel electrophoresis provides information about size and integrity of RNA, but it may miss RNA species of the same length but without poly-A tail, while IPRP HPLC method also provides information about integrity and allows for efficient detection of RNA without tail. Likewise, size exclusion chromatography again provides information about RNA integrity, but at the same time, it may detect larger mRNA aggregates and also small molecule impurities such as residual NTP. So you see, each of these procedures gives a critical piece of information about RNA quality, and then all these pieces combine together to reveal a whole picture about the overall quality of RNA.
Hannah, Ben, would you like to add anything to that?
Yeah, and I'll just add, as somebody who likes research and development, there's a lot of development and new aspects to all of those types of assays. So seeing the pace of new ways to measure cap or new ways to measure the integrity or new ways to measure, I think it's a really exciting area of the field. It's just so many new things to characterize and orthogonal techniques to apply. So like I'm sure Hannah would agree, you really need to nail down sort of what aspects you want to prioritize and then design your key quality attributes, key critical attributes after that.
Yeah, and you can keep in mind too that throughout the development process, you can always add on more analytics as you go. And so one or two tests may be sufficient early on to see how your mRNA is characterized for any of your key quality attributes. And as you build out your clinical pipeline, you can introduce more tests so that you have that redundancy.
Very good. How do current identification methods for mRNA differ from each other? There seem to be a lot. Which one would you recommend? Inna, let me start with you again.
I think that the choice of the proper ID method is primarily determined by the phase of development. For example, next generation sequencing is very helpful at the early discovery phase when screening for hundreds of RNA constructs simultaneously. However, at the later stages, when once the mRNA drug candidate construct is locked, Sanger sequencing can be a better choice as it is still a golden standard in industry with clear path to method qualification and validation, which becomes very important at later stage of drug development. And it could be an issue with next generation sequencing. An orthogonal approach to mRNA sequencing, which greatly evolved in the last few years, is fingerprinting/mapping of RNA by LC-MS/MS. In this approach, mRNA is first enzymatically digested to smaller ribo-oligonucleotide fragments, which are chromatographically resolved, and then their identity is established by LC-MS/MS. One advantage of the LC-MS-based approach is that it may potentially allow to gather information about RNA sequence, 5' cap, and tail within the same experiment.
Ben, let me come back to you. How does the intended therapeutic application of the mRNA guide the choice of design and analytical methods?
Yeah, so like I said earlier, RNAs come in all sorts of flavors, even though they have, in general, the main components all the same. There are ways to modify each of those components to greatly affect what the attributes that you want to put into your modality. So what everybody's probably most familiar with, we'll just say start with a vaccine. The idea of a vaccine is you want to stimulate the immune system against a particular antigen or multiple antigens. And so in that setting, you would prioritize robust protein production and a strong but not too strong controlled immune response. So you'd want RNAs that put an emphasis on cap structure, on longevity of the RNA. You would want a poly A tail length and UTRs that have been optimized for your use case and for your antigen or your protein to deliver the longest expression and the most robust response. For another example, for another type, for example, maybe gene editing. So for CRISPR-Cas9, kind of the most well-known sort of gene editors, you might require two RNAs, and those would be different types of RNAs. So one might be designed to produce the nuclease, the Cas9 protein. So that would be a high protein production type of RNA with caps and UTRs and poly A tails that would be designed to drive expression, protein production, and maybe have modified bases that would be designed to reduce immunogenicity in that case. And then you would also have a separate guide RNA or something like a sgRNA that would have no cap, no poly A tail, but might have modified bases such as 2'-O-methyl to promote stability. So there's a lot of great examples in the literature of having very specified modified bases and guide RNAs. Another type of modality that's pretty gaining a lot of traction now is very exciting, cancer immunotherapy. So using cell therapy, but using RNA to deliver that direction for the immunotherapy. And in that case, the whole goal of that, instead of using, let's say, a viral technique to introduce your gene of interest, you introduce it with RNA, and so you're relying on the transience of the RNA, and you're designing your therapy such that you can introduce a very specific target. For CAR T, for example, you might introduce a chimeric T receptor, but you won't want it to last forever, and so you'll design your RNA and tweak your cap and your UTR and your poly A tail such that you get good response, good production, but maybe it doesn't last for years and years and years like a virus would. Protein replacement therapy is sort of kind of piggybacking on the lines of vaccine. You want strong protein production, so all of your focus is going to be on designing, robust response, high yield. But in this case, you probably don't want a really strong immune response. And that's because you might be injecting this multiple rounds. You don't want to develop an immune reaction to the RNA itself or to the lipids that might be involved in the formulation. And so in that, you really want to tailor, and we didn't cover any of the lipids or LNPs in this topic, but just something to keep in the back of your mind. And then a new and really exciting area of research, kind of still up and coming, structured RNAs, so RNA aptamers, which are RNA molecules designed to fold into a specific secondary and tertiary structure, or tRNAs, which have a very well characterized structure. tRNAs are being evaluated now for stop codons, where you have aberrant stop codons, and you want to sort of allow a protein to be read through. There's a lot of diseases that can be aided with that, but those types of RNAs are very structure dependent, and you probably won't have a cap, you probably won't have a poly A tail. It's going to be your whole design of the RNA is going to be around how does it look, how can we manufacture it such that it always looks the same. So sometimes you'll have modified bases for those, like I said, no cap or poly A tail, and in that case you'll have very specific defined modified bases. tRNAs, for example, have dozens of modifications at many bases.
Great. Can you talk about how these different uses impact the design, manufacture, and quality attributes of the RNA?
Sure, Hannah, can you take that one for me?
Yeah, I got this. So I think, when you're assessing what the right manufacturing design and quality attributes are for your application, you want to start by really looking at, to what Ben was alluding to, some of the key players in the response that you're looking for in your patient population. So, for example, if you're looking for low immunogenicity, a therapy such as a protein replacement, you want to start by saying, okay, we're going to need low double-stranded RNA. We need to optimize that mRNA to elicit the lowest possible immune response. Low double-stranded RNA is something you can actually design a synthesis reaction upscale or upstream of your downstream purification process to perfect. And that's the philosophy that is kind of the strongest when you're approaching your manufacturing design. If you can get your synthesis reaction to really highlight the key quality attributes and select for those before you get into complicated purifications, your downstream processing will be simpler and your mRNA will overall be easier to manufacture and of a higher quality. So double-stranded RNA, ideally you're optimizing at the synthesis. If you really need to purify for that, you can introduce some challenges with methods such as reverse phase HPLC. Wherever you're selecting for fractions that don't have a clear quantitative cutoff for take it forward or don't take it forward, you can run into some compliance issues with how are you going to define what you're actually going to pool into that final manufactured product. Scalability is another huge concern for some of these, especially when you're talking about an application like a vaccine, where you need to dose a large population. So there you can optimize your synthesis reaction to achieve a high yield, and then pick purification strategies that are going to be able to handle those kilogram quantities downstream so that you're not dealing with a month-long purification project for your clinical batch. I think the most important kind of takeaway on your manufacturing strategy, and it plays into a lot of what Ben's been talking to as well, is one size does not fit all. Even in the same application, your requirements, your optimal design is going to differ by the construct or by the target and how you design that mRNA to get that perfect response in your patient population.
Very good. Thank you. Okay, we've covered how the specific therapeutic application impacts design and manufacturing. Inna, let me come back to you. How does the intended therapeutic application guide the choice of analytical methods?
Well, regardless of the application, mRNA characterization and analysis will encompass the same critical quality attribute as we previously discussed. However, the number of orthogonal tests, as well as the level and depth of certain assays, may differ depending on the application. I will use the same example as Hannah just used, double-stranded RNA. Double-stranded RNA species must be very low for certain applications such as cell therapy or protein replacement, and therefore I imagine that a very specific and potentially sophisticated analytical procedure must be developed to ensure a low level of double-stranded RNA. On the other hand, double-stranded RNA may not be as critical for vaccine application, where double-stranded RNA might even help serve as adjuvant. Therefore, in this case, a simple limit test using dot-blot immunoassay might be sufficient to confirm, say, batch-to-batch reproducibility of mRNA. Again, another example could be functional assay, where application determines the type of functional assay necessary for mRNA. For example, therapeutic applications such as protein replacement may require a sophisticated cell-based assay or even assays to confirm not only expression of the target protein, but also its full functionality. While for the vaccine application, in vitro assay demonstrating efficient expression of protein might be enough. Therefore, I think it's up to the drug discovery and CMC team to determine what critical quality attribute of RNA drug candidate they need to evaluate and to what extent. And this information will help determine if analytical methods are already available or they may need to be developed.
Very good. Well, this has been a fantastic overview of this field. And in closing, I'd like to go around the room and ask all of you, what are you most excited about for the future of mRNA-based drugs? Ben, I'm going to start with you.
Sure. It's a great question. And in R&D, it's very apropos. So to me, the most exciting aspects of RNA-based drugs are where they intersect with other therapeutic modalities. So it's great to see RNA having a foothold or toehold in lots of different design spaces. So one example would be using, like I mentioned earlier, using the transient nature of RNA for cellular reprogramming or for cell therapies, where you want a response that's tailored. You don't want it to last forever, but you want to have a controlled off rate. Another really exciting area of research in the other end of it is circular or self-amplifying RNAs, where your focus is on increased duration, and you're really trying to find ways to make the RNA last, not necessarily forever. Maybe that would be ideal in some circumstances, but for a much longer time. So there's just lots of ways to design your RNA to alter its functionality and purpose.
Great. Hannah, what are you most excited about?
Yeah, so, I love this field. I think it's fascinating how it has grown, especially with how through the COVID pandemic specifically, certain aspects of the technology had to grow very quickly. And now we're kind of playing catch up in some of the other areas. So I am the most excited to see how the innovation on things like automation and solutions for key manufacturing challenges are catching up to the field to really help speed up that pathway from discovery to helping patients.
Very good. And Inna?
Well, one thing I'm excited about is the emergence of new therapies for a broad range of diseases, including cancer. But apart from that, I'm very excited about the new analytical technology for analysis of mRNA that are being developed as we are speaking. And I'm very excited about learning these new technologies and also about the potential opportunity to personally contribute to the development of new analytical methodology for therapeutic R&D.
Wonderful. Well, thank you so much for your great your expertise, clearly, and this very enjoyable conversation. I'm sure the audience got a lot out of it. That just about does it for today's webinar on mRNA therapeutics. I want to thank all of my guests today, Ben Hudson, Hannah Ellerbrock, and Ina Kukareva, all from TriLink Biotechnologies, doing great work. TriLink, of course, for sponsoring today's discussion, and the Gen Multimedia Group for producing today's webinar, led by Liana Jabs and our friends at Workcast. Thank you also for your questions. If you put them in, we're not taking questions live right now, but somebody from TriLink will be getting back to you shortly. So thank you for your interest in today's subject. So all that remains for me to do is to say thank you for watching. I'm Kevin Davis on behalf of everybody at Gen and the CRISPR Journal and Gen Biotechnology and the scores of other journals that we publish related to the RNA field. Thank you so much for watching, and goodbye for now.